Okay, with this paper, you have to cover the things related to the ATP and enzymes. Okay, you know, the lesson number 2 can be divided into two main parts, cellular basis and chemical basis. Under the chemical basis, there are some topics. First one is elements, and the second one is compounds. Okay, under the compound part, we are talking about both inorganic and organic compounds. That means water, especially water as the inorganic compound. And we have to discuss 4 major organic compounds: carbohydrates, lipids, proteins, and nucleic acids. And other than that, enzymes and photosynthesis, cellular basis. Like that. So now we are here with enzymes. We are talking about ATP part. Okay. First one, correct about ATP. What about the structure of ATP? What are the 3 main components of ATP molecule? This is the ribose sugar. And what is this nitrogen base? What is the name of nitrogen base in ATP?
Adenine.
Yes, very good. Adenine base. And how many phosphate groups are there?
3.
Yes, 3. And how many high energetic bonds between phosphate groups? How many high energetic bonds?
2.
Yes, very good. 2 high energetic bonds. This is not a high energetic bond. Actually, this is the first carbon, second carbon, third, fourth, and fifth carbon of this ribose sugar. This is a pentose sugar. So this is not the high energetic bonds. Therefore, 2 high energetic bonds, 3 phosphate groups. Okay, phosphate group, adenine base, and pentose sugar. It is the ribose. Pentose sugar is ribose. Nitrogen base is adenine. Both adenine and sugar group are considered as adenosides. This part we considered as adenosine. Adenosine 1, 2, 3. Adenosine triphosphate. ATP means adenosine. Adenosine means the pentose sugar group plus adenine group. Adenosine. Tri means 3. Triphosphate. So after removing the terminal phosphate group, we can take the molecule what? After removing this part, how we can call this molecule?
Adenosine diphosphate.
Very good. That means ADP. No? ADP. ADP bound with inorganic phosphate group and produce ATP. So both adenine and sugar group are considered as adenosine. A phosphate group is attached to the 5th carbon of pentose sugar group. After hydrolyzing this one, after hydrolyzing ATP, that means adenosine triphosphate, we can take ADP and PI, that means the inorganic phosphate group. All are correct, no any answer. No any answer here. So, can you understand, Akila?
Yes, yes, miss.
Okay, not a cofactor. So, as you think, what is the answer? Not a cofactor?
ATP.
Very good, ATP. Cofactors are 2 types. What are they, Tinashi? Cofactors are 2 types.
Inorganic and organic.
Yes. Inorganic cofactors and organic cofactors. Organic cofactors are 3 types. What are they? Vitamin derivatives, FAD, NAD plus, and biotin. Inorganic cofactors, zinc 2 plus, iron 2 plus, and copper 2 plus. So you can make a small note in this space here. Okay, this one. And answer is number 4. Incorrect about enzymes. Enzymes increase the activation energy requirement. Correct or wrong? Activation energy requirement.
Wrong.
Wrong. Why? How we can say it? Enzymes increase the, or how we can say this statement?
Enzymes decrease the activation energy requirement.
Yes. So, Tinashi, can you tell me what do you mean by the activation energy for any reaction, any chemical reaction? Activation energy means what?
The energy required to catalyze or do some.
Yes. Actually, here we will think about sucrose hydrolyzing reaction. That means sucrose plus H2O into what are the products here? What are the products? Glucose
Plus fructose.
Yes, fructose. Enzyme is sucrase. Sucrase enzyme. So here the enzyme is catalyzing the reaction. Catalyzing. Catalyzing means decrease the activation energy. Decrease in activation energy, that means increase in the enzymatic reaction rate. No, enzymatic reaction rate. Actually, when we considered reactants like A and B, I think nitrogen and hydrogen produce ammonia. So what are the bonds between nitrogen? It's look like this, N2 and H2. Initially, we have to break these bonds, okay? Break the bonds within the reactants, and finally we have to form new bonds like this. So activation energy means the energy required for breaking these bonds in the reactants. It is the activation energy. These biological catalysts or enzymes decrease the energy. Okay, decrease the energy for breaking these bonds between the reactant molecules. So, catalyzing means decreasing the activation energy, then increase the enzymatic reaction rate. So, I think you got it. You both got it. Akila, are you okay?
Yes.
Okay. Enzymes increase the activation energy requirement. So, it's wrong. It should enzymes decrease the activation energy requirement. They are substrate specific. Correct or wrong? Substrate specific?
Correct.
Correct. So, yes, actually. So, Akila, can you understand Sinhala? Any
Yeah.
term? Okay. Sucrose here, substrate specific means ඒ කියන්නේ දැන් මේ enzyme එකෙන් catalyze කරන්න පුළුවන් මේ substrate එක විතරයි. Okay, that is we call that substrate specific. Can be occurred. ඒ කියන්නේ දැන් sucrose කියන substrate එක catalyze කරන්න sucrase enzyme එකමයි ඕන. Maltase enzyme එක use කරලා we cannot catalyze this reaction. So sucrase enzyme is specific to sucrose substrate. Can be occurred in living cells. Yes, enzymes are biological catalyst. Rumana, now we are in question number 3. Okay.
Okay, teacher.
Okay. So question number 3, answer number 3 also can be considered. Many enzymes are globular proteins. Yes, many enzymes. You can highlight this term. Many enzymes. No, it should not all enzymes. All enzymes are not globular. Okay, all enzymes are not globular proteins. Many enzymes are globular proteins, so it can be considered. Many enzymes are heat unstable. So how we can explain this this statement by using a single term. How we can explain this? In heat unstable means what? How we can explain? එක term එකක් තියෙනවා නේද මේක මේ heat unstable වෙලා කියලා කියන්න පුළුවන් එකක්. කොහොමද heat එක ටික ටික increase කරගෙන යනකොට මොකද්ද වෙන්නේ? enzymes are actually proteins. So when we increase the temperature or heat, what would be happening to the proteins after passing about 4 degrees Celsius?
Denaturated.
Yes, enzymes are made up of proteins. When the temperature is too much high, these proteins can be denaturated. So denaturation means denaturation එකක් කියලා කියන්නේ දැන් protein එක ගොඩක් කලාවට tertiary structure එකනේ කියන්නේ protein එකේ තියෙන tertiary structure එක තමයි මේ enzymes වල include වෙලා තියෙන්නේ. එතකොට tertiary structure එක build up කරන්න contribute කරපු intermolecular interactions ඔක්කොම break වෙනවා upon the heating. Then enzymes are denaturated.